I think we can start this webinar of the series of Exploring SRF beamlines. Today is the second webinar for a beamline dedicated to structural biology, and it's my pleasure to introduce Igor Melnikov, who is in charge of ID30A-3, also known as massive number 3, the mini-focusing beamline for macromolecular crystallography. Igor, the stage is yours.
Thanks, Adriana. Thanks everyone for joining. So today the topic is ID30A-3, also known as massive 3. Let me start from the overview of beamline layout. So massive 3 is one of the branches of massive line, which is in fact is a part of ID30. And ID30 is built in a content configuration. It has two undulators, U21.2, which gives beam to ID30A or massive branch, and U35, which gives beam to 30B. They all share same optics hatches with 30B beam just passing through in a straight pipe, passing through the optics components for massive branch, which are in the optics hatch 2. So massive branch has two, massive line has two branches, massive 1 and massive 3. And the optical setup is the following. The wide beam, firstly, is focused by the compound lenses, is vertically focused by the compound lenses. Then it reaches monochromator for massive 1, giving a monochromatic beam. And then it passes through massive 1 monochromator, goes downstream. There is no massive 2 anymore. And it reaches finally silicon crystal of massive 3, which gives 111 reflection, monochromatic reflection further downstream to the experimental hatch, where it's focused by the KB mirror horizontally, and the KB mirror is already quite close to the sample position, in fact. Overall, this gives a beam at the sample position roughly 15 micron in size, and it's being collimated and cut by a set of apertures. Typically, we use 15 microns and 10 micron apertures. And the beam focus is horizontal focus is located at the position of the detector, which reduces the diffraction spot size. The experimental setup for massive 3 is a very typical MX setup. It consists of MD2 diffractometer, a very fast Eiger detector, and the flex sample changer, which consists of a robotic arm and its storage dewar. So in a standard MX experiment, protein crystal is located in a loop, is mounted in a loop on a pin and exposed to the X-ray beam. So producing diffraction, of course. And the design of MX experiment implies that sample must be diffracting at different orientations, producing different diffraction patterns. In such a way, because each diffraction pattern is in fact a subset of a series of intensities. So taking patterns at different orientations, we obtain a full set of this series. And the thing is that the final structural data quality is very much dependent on the completeness of these series of intensities. And in fact, that's why the crystal must be rotated and collected as much as possible rotational data. For that reason, the crystal must be properly centered in the beam during rotation. That means that centered, all the 3D coordinates of the crystal must be put into the intersection of the rotation axis and the beam. Very common way to center the crystal is using X-rays itself. So in at the ESRF MX beamlines, we typically do a mesh scan, X-ray mesh scan in a predefined area, producing diffraction images, and after the scan, these diffraction images are analyzed, and we can construct such a diffraction map, or with further analysis, a crystal map. And using this map, we can center the crystal, but only in 2D, so only two coordinates are known. To get the third coordinate, usually the crystal is rotated by 90 degrees, and an additional line scan is made to get the third coordinate of the crystal. This works in 80 to 90% of the cases, but in very difficult cases, it can fail. Like if you have several crystals in a loop, or diffraction is not homogeneous, or anisotropic. For that, there is a recent development at the ESRF called double mesh. So instead of the line scan, we do a second mesh with in some angular difference. And the problem previously was that it was not possible to distinguish between different crystals in these two meshes. But now, thanks to some really fascinating method, we can see and select different crystals, different same crystals in these two meshes, and properly center each of them. As you can see here in the example of bacterial rhodopsin, 7 positions were selected, and at these positions, 60-degree wedges of data at each position was collected. And in fact, this it could be meshed together, but in fact, these 60 degrees have a complete produce a complete data set, because thanks to the symmetry of the crystal. But sometimes the crystals could not be properly centered in the beam, or they are susceptible to radiation damage very much. And in these cases, there is a method of serial crystallography, where a complete data set can be obtained by merging together wedges obtained from different crystals. A single wedge can be as small as a single image, even not rotational image or still image. But on Massive 3, we adore our favorite methods, mesh and collect, to do this serial fashioned data collection. Basically, what we do is we do a mesh scan over the whole large crystal area, and we go back to each diffracting position, and we collect small wedges. typically 5 to 10 degrees. And those wedges are then merged together, selecting the more isomorphous ones. Sorry. And the method is really nice because it allows much better accuracy in intensities and in error estimation compared to still image methods, because we collect most of the data is full intensities and non-partial ones, and we can do cluster analysis and select isomorphous data. For that type of experiment, we designed a fixed target sample holder for Massive 3. Is basically a 3D printed single hinge clamp, and sorry, in this clamp we put a Mylar foil sandwich with the crystals dispensed in between this sandwich, and it looks like this. And in fact, this sample holder is very useful for our time resolved studies, which I will be talking in the next slides. So that's the setup for time resolved on Massive 3. We are using, it's quite simple. So first of all, we use a laser with a very fast shutter, and the laser lights cut by the shutter is then collimated to the optical fiber and transmitted to the objective, and then is focused on the sample in a spot size of roughly 100 microns. And the simplicity is in that we have a trigger signal from diffractometer to the detector to start the acquisition, and we hijack this signal, and using Opium card we process it and convert to laser shutter signal, and by that we can obtain laser pulses in the very beginning of data acquisition, limited to several milliseconds. So typically we use 1 millisecond or 2 milliseconds exposures of laser. And that setup we are using in for the time resolved serial oscillation crystallography method. So basically we are using a mesh and collect. We go position by position to each crystal, and we collect small wedges, 5 degrees. And each data collection starts with a laser pulse, which starts the reaction. Here, for example, we had bacteriodopsin again. Laser pulse initiates the reaction, and thanks to the fast Eiger detector, which with a minimum acquisition time of 1.4 roughly milliseconds, now each image corresponds to a certain time step in the reaction. And by collecting multiple data sets, we can take the images corresponding to a certain time step and merge them together. Take them from all the data sets and merge them together and obtain the complete data set. And finally, we can merge from different time steps and obtain structures, solve the structures corresponding to these time steps. This is, of course, very much dependent on the numbers of data sets you acquire. And the more data sets you get, the more temporal resolution you have, which is limited basically by the minimum acquisition time of 1.4 milliseconds. You can take as little as one image from each data set, but you will need a lot of data, of course. And here's example study of this time-resolved SOX. We collected roughly 150 data sets in a mesh and collect fashion from a loop, and selected 119 of them to be isomorphous. So we merged them together, and then each data set was cut into five parts, temporal parts, and each part corresponding to 15 millisecond time step. And here you can see some results. So electron density difference map between the ground structure and the 0 to 15 millisecond time step structure in the reaction. You can clearly see some structural differences here. Now we collected already more than 1,000 of data sets recently, and this allowed us to get to the temporal resolution of a couple of milliseconds, which is quite incredible for this simple setup. So another notable development here on Massive 3 is the internal ESRF development, is the compact UV visible microspectrometer, which can be mounted on axis directly and measure spectra simultaneously with X-ray exposures. This is done with great support and close collaboration of ICAS lab in the ESRF. Here you can see the spectrometer mounted on axis. The excitation light comes from the bottom objective. It's basically a white lamp light. It's focused on the sample in a spot of roughly 25 microns. Can be less by using less fiber diameter. And the transmitted light then is collected through the second objective and sent to analysis by a spectrometer. So here's some example study of the spectrometer. These are rhodopsin crystals dispensed in this our favorite milfoil sandwich in a clamp, and these crystals were subjected to radiation damage. And you see here that the retinal absorption peak, it fades out with the taken X-ray dose, and also it shifts to the right. Finally, I mentioned some recent upgrades of the beamline. The detector got a new upgraded machine. We are now also up to date very much with MXQ4, and the maximum achievable resolution has been extended tremendously to 1.17 angstroms now for our small Eiger detector. That's really great. So overall, to summarize, Massive 3 shares high operational standards and unique methods across the fleet of structural biology group beamlines at the ESRF. As all the beamlines, it has some own features which are for Massive 3 in serial data collections, time-resolved SOX, and spectroscopy, basically. let me thank you, structural biology group members, and also ASRF staff for the support, and also for the huge support from their side, the MBL instrumentation team. So with that, thanks very much, everyone.
Thank you very much, Igor. Please write your question in the question and answer box. I forgot to tell you. So while we wait for questions, I have one. Which is the highest difference between what you could have done before and what you can do now with the extreme brilliance source with respect to time, but also with respect to radiation damage of the crystals? I mean,
well, it's a good question, but in fact, there was not much of a difference for Massive 3 because the beam was still bright in the previous ESRF setup. We got slight increase in flux, and of course there was increase in the divergence, but we did not notice it, let's say. So we keep the beam 15 microns and with relatively high flux of 2 to 10 to the 13 photons per second. And in fact, even for one millisecond exposures, that's already too much sometimes. So we need to attenuate the beam.
yeah, that would have been my second question. Do we need to attenuate the beam?
Yeah, especially because all these time resolved studies are basically are room temperature studies, of course. That's why we cannot afford this much of a dose on a sample. So that means we need to attenuate. During these TR-SOX studies, we usually use 5 to 10% of the beam.
It makes sense. Another question is, when you do your mesh and collect, which is the minimal size of the crystal that you would like to have in your Mylar film? Will you reckon to have something like 10 to 20 micron or smaller, so such that it's more of a slurry of crystals than real crystals?
So ideally, that would be 10 to 20 micron crystals, a lot of them distributed homogeneously. But I think depending on the diffraction quality, which is of course reduced for small crystals, can be even 5 microns.
Thank you. And how do you mount your crystal in your Mylar, or do you just do the crystallization directly in your little chip?
Yeah. So one option is to do crystallization. We are working with membrane proteins, so we can use just crystallization in syringes and directly dispense the crystallization slurry inside the sandwich. Or I did opening of crystallization wells and transferring crystals directly to the sandwich. It's not a big deal, but of course it takes time. And the plan is now to set up a crystallization directly in this sandwich and just cut it directly from the crystallization plate and put on axis. That would be the simplest thing.
Thank you. There is one question online. When sending a dual to ESRF, which is analyzed at ID30A, could we use TR SOX for any of the sent crystal, or there are special conditions?
Any of what? Sorry.
Can we use TR SOX for any of the crystals which have been shipped? So analyze structural dynamics at room temperature, or are there special conditions?
Yes, there are special conditions, of course. It depends how you initiate the start of reaction in your crystal. For us, we have this bacteriorhodopsin, where you initiate the reaction by the pulse of light, and the reaction is reversible. That means that if we expose too much of a sample area, then in a couple of seconds, all the crystals will be back to ground again. This is an issue, of course. So that depends. I think we can come up with some mesh type containers where we can expose one window at a time to not touch any other crystals with the laser pulse, I mean.
Thank you. Maybe Mila Lutold can write to you directly
Yeah.
to explain the system.
Yes, please write directly to me.
Maybe I missed it, but which is the diameter of your laser beam which hits your crystals?
It's now roughly 100 microns.
Okay. So, I mean, it can, if given the size of the crystal, it will illuminate all your mesh. So
Not all, but a couple of neighboring crystals, of course. But there is a development. There is a development to put the to send the laser to send the light beam from the on-axis viewer. So we'll see how which size spot size it can give. It can be less, but the spot size, but you lose the power with that.
Okay. So for the time result studies, you would reckon to have a system in which the K on or K off rates are in the millisecond or longer times in order to be able to cut your slices.
Yes.
So not very fast reactions.
Yeah, that's actually fine for most of the rhodopsins, which have a photocycle of typically 100 milliseconds, maybe.
Okay.
So that's an ideal situation for that.
Absolutely, yes. Not so fast.
Yeah, of course, not for the early intermediates of the reaction, but
yes, of course. But at least you have a wide range of reactions that can be nicely to study for enzymatic reactions in the slow period, if a millisecond could be called slow. Please, if you have other question, please don't be shy, write in the question and answer. Otherwise, we can stop the recording